Very good morning, everyone. I'm really excited to talk to you about the applications of nanotechnology in animal agriculture. Myself is Saji George, and I'm an associate professor at the Department of Food Science and Agriculture Chemistry, McGill. Before I start with my presentation today, I would like to thank the organizers for having given me this opportunity, and I hope this will lead to some collaboration with the stakeholders of interest. My group at McGill studies nanomaterials for the potential toxicity, understanding the mechanisms, as well as studies nanomaterials for developing exciting applications in diverse food and agriculture sector, and to work with industry to translate those exciting findings from the lab. So without much time, let me talk about what is nano and how that nanotechnology can be applied, giving some examples from our own research. So by definition, nanotechnology is the manipulation of matter at the nanoscale to generate size-dependent functionalities that are different from atom or the bulk counterpart. So most of the materials, for example, gold. We know gold in atomic form or molecular form, as well as gold in bulk form, which is the gold coin or a block. There is a state in between, that is the nano, where the surface area of the material is extremely high if you compare it with a bulk material. And also it has a high percentage of surface exposed atoms in comparison to a bulk material. And the surface is extremely smooth so as to give a very high hardness, for example. And as you can see, the state of this matter is between atom or a bulk counterpart. So for simplicity, you can think they are actually in between Newtonian physics or quantum physics. And as a result, there is a dominance of quantum effect. All these make nanomaterials new and exciting and give them novel properties. And that applies when they interact with the biological system as well. So when nanomaterials go to interact with the biological system, their size, their shape, their surface chemistry, their core chemistry, etc., actually determine the type of interaction, whether it is more beneficial or hazardous. For example, the size, nano size, because they are extremely small, they can trespass many of the biological barriers. And given their shape, the hydrodynamic properties, as well as their flowing when they flow through the circulatory system, they can be different from a bulk material or an atom or a molecule, how it get transported in the circulatory system. The surface chemistry can be manipulated to make it more targeted. Their core chemistry can be manipulated to have multiple entities. So another possibility is multifunctionality. You can actually bring in different functions into a single entity in order to make a material that is multifunctional in nature. All this make nanotechnology exciting, and it has applications in diverse sectors, including food and agriculture, where it is applied for infection control in farms, feed and nutrition manipulation to deliver micronutrients, for example, rapid disease diagnosis, vaccine delivery, pest control, health monitoring. All these are exciting new applications of nanotechnology where there are industry players who are providing solutions based on nanotechnology for diverse challenges in animal agriculture. Now, I will give few examples from my own research. The first one is a research that we conducted in Singapore, where we developed a nano vaccine to protect a farmed fish. Here, the fish that is seen is a sea bass. When this fish is farmed, they grow in high density. As a result, there is a high chance of disease transmittance. And if there is a disease strike, within 2 or 3 days, the entire population can be lost. And that was an issue with a farmer who came to us. We were able to identify what is causing the disease, which was a tropical bacterium called Tennessee bacterium aritanum, which causes the devastating disease. And our colleagues were able to identify the disease-causing bacteria, grow them in laboratory conditions, inactivate the bacteria, and we were able to inject it into the fish bellies. So the fin pellets were, when they were exposed to real challenge, they survived. There was more than 90% survival rate as a result of that vaccination. But only one problem. Each fish has to be knocked out and then it has to be injected with the vaccine. So we rationalized that instead of using a microscopic level of vaccine needles, why don't we actually devise a nano needle where the vaccine components can be kept inside the needle? And this one, when it dispersed into the water when the fish is growing, it would get self-injected into the bellies as well as into the gills so as to elicit a good immune response. So we screened a library of natural nanomaterials, including clay, silica, chitosan, as well as hydroxyapatite, and combinations of thereof. And we screen for the safety. That's the first and foremost thing in fish. And clearly you can see none of them have a very high safety signature, except for the paucity control, which is a silver nanoparticle. Same thing can be seen in the zebrafish embryo, which is used as an in vivo model for many of the toxicology studies. And we also studied the toxicity in human cell line as a proxy for a safety issue later on if you were to ingest this nanomaterial, which is included in the fish. We also studied the efficiency in terms of delivering a protein. Here, what you're looking at is the delivery efficiency of green fluorescent protein. The rationale is if it can deliver GFP, which is a green fluorescent protein, it may be able to deliver the vaccine component. As you can see, among the tested ones, one of them actually showed a higher efficiency in terms of delivering the vaccine, the GFP into the cells. And we also tested it in the fish system, and you can clearly see in the fish embryo here, you can see the signatures of GFP in the gut as well as in the gill area, which we actually confirmed using ICP-MS, taking aluminum as a proxy for the nanomaterial that we used, and most of them were found in the gills and the guts in the first week after the exposure, and clearly it resides to the normal level at the fourth week, and the minimal presence in the edible portion of the fish, which is the muscle portion. And we compared their performance with the toxicity or safety or the cost to identify which is an optimal delivery platform, taking into consideration of these elements of sustainability. And obviously, we wanted to choose the one which is in the blue area. So we make sure that it is sustainable, not only in terms of its safety profile, but also in terms of the cost. And we tested that system for delivering outer membrane proteins from the bacteria that causes the infection. And then we found that the nano system is in fact capable of eliciting a better immune response in the fish, and also the gene expressions were studied. Now this technology is patented and it is now taken by the farm with whom we were working, and they are applying it for their fish production. Another example that I will give is from the study that we concluded just a few months ago, which is about the antimicrobial resistant bacteria isolated from the swine farms in Quebec. As you all know, most of the antibiotics that are generated, around 70% of them, actually is used in animal agriculture to ward off infections and also to promote the growth. And as a result, the farms become the birthplace of many of the antimicrobial resistant forms of bacteria, and then they can spread to public either through food channel when we consume a food that is contaminated with antimicrobial resistant bacteria, or directly from the farm through the workers, for example. Either way, it is a problem not only for the farming industry but also for public health because of the huge cost involved, as well as disease prevalence in terms of loss of life. We characterize a small library of Staphylococcus aureus as well as Salmonella that were isolated from swines and that was in collaboration with Dr. Mary O'Dale from University of Montreal. And these bacteria were characterized for their antibiotic resistant using a panel of antibiotics that are usually used in farming. And we identified all of them are multi-drug resistant, and especially look at this Salmonella typhimurium, which is resistant to almost all of those antibiotics that we tested. And we identified this bacteria that is a peculiar one. It actually has a high cell proliferation in presence of ampicillin. You can clearly see that in presence of 250 or 500 microgram per ml concentrations of that ampicillin, the proliferation is higher in comparison to the control, which is not exposed to any. We are doing more research to understand the mechanism involved in here, because it's a, like I mentioned, it's a peculiar observation about a bacteria that proliferate highly in presence of antibiotic. We identified the mechanisms that are prevalent in these bacteria, and we found that it is because of a flux pump activity as well as beta lactamase activity measured through the standard protocols established. We wanted to identify this mechanism operational because if we were to design a drug, we would like to see what exactly is the mechanism so that we can actually make those molecules available in the system that we develop. Now, we did a checkerboard assay, which is in essence a combination to check the effect of combining more than one drug together and to see what kind of effect each drug has on the other one, whether it is an indifferent, which means no effect, or an adverse effect of each other, or it's an additive effect, or an even better synergistic effect. So we wanted to identify the molecules that give additive effect. So here is a list of adjuvant molecule, what we call, because they are flux pump inhibitors or beta lactamase inhibitors, or molecules that facilitate the uptake of the antibiotic into the bacterial cell, for example. Here we took Salmonella enterica as the bacterium that showed the highest level of antibiotic resistance. And we tested against the panel of adjuvants and primary antibiotics. And then we could see that some of them exhibited synergistic effect. For example, ampicillin with tazobactam, which is an inhibitor of beta lactamase enzyme. And another exciting molecule combination was tannic acid and silver, clearly showed a very good synergistic effect. Now, our effort was to bring these two together in a nano material. Why nano? Because nanomaterial, as we discussed before, provides multiple benefits in terms of its tissue penetration, providing stability to the cargo, and also possibility of targeting specific tissue, for example, as well as combinations, possibility of delivering more than one drug together at a go in a single entity. So we generated silver nanoparticles that are grafted with tannic acid, for example. Here you have silver as well as tannic acid. And another system where you use a hyaloicide clay needle-shaped particle where it's loaded with tannic acid and surface modified with tannic acid, and then graft that surface with a silver nanoparticle. This is an in situ formation of silver nanoparticle onto the surface of a hyaloicide clay grafted with the tannic acid to generate what we call as a composite material. We characterize this material for the presence of these entities, especially tannic acid and silver, and we were able to generate and successfully characterize, and this will look like this. Now, we tested for the presence of silver and tannic acid, as well as the delivery or release kinetics of these two entities, and we can clearly see that system do have silver as well as tannic acid, and then you can also see the delivery is slow and sustained, especially in the nanocomposite when we look for silver or for tannic acid here. As a result, if you're testing for its minimal inhibitory concentration in bacteria, here is the result of that test in E. coli or SRS. Those are two quality control strain, and then the multi-drug resistant Salmonella isolate that we had discussed before. Clearly, that composite material showed much improvement in the MIC value, minimal inhibitory concentration value of these drugs. And so we tested this also in a small organism model. Here we took the intestinal infection model of a C. elegans, which is infected with the salmonella, and then we look for their lifespan. We compared it with a non-infector. Obviously, the non-infector one survived more than 20, approximately 28 days, while the infected one, when it is not treated with anything, has much lower survival rate. Obviously, the salmonella kills the C. elegans, that's the worm. When these infected ones are treated with a 62.5 microgram per ml of concentrations of the nanocomposite material, clearly we see a tremendous improvement in the survival of this animal, this worm model, the in vivo model. And it shows that possibility of circumventing the infection by a multi-drug resistant bacteria in an intestine. And future studies, we will conduct more studies to understand if we can target the intracellular pathogen and eliminate it, and also specifically target and kill the pathogenic bacteria, leaving behind other bacteria, and then showing this benefit in real animals, especially in swine or poultry. We are looking forward for partners for those kind of studies. With that, I would like to conclude my presentation, and I would like to thank the real heroes behind this work, which are my students and all the wonderful collaborators I have who actually, with whom we study multiple aspects relating to nano, and obviously the funding agencies for enabling all this wonderful work. And thank you one and all for listening to my talk. And I will be happy to interact with you at the symposium. Looking forward to that. Thank you.